Does an allergen carrier always contain major, intermediate and minor allergens? Natural allergen carriers, such as grass pollen, always contain all allergens, ranging from major to minor allergens. These allergens have natural variations, for example, in the concentration of the individual allergens. All specific allergens are capable of triggering sensitizations in an allergic patient. Therefore, it is crucial for diagnostics and therapeutics to optimally represent the natural source containing all sensitizing allergens. To ensure this, the manufacturing process and the procedures used are crucial. Any allergens that are lost during the preparation phase of the raw material or during production cannot be added later to the extracts, which, by the way, is the basis for the commercially available products. The production processes are manufacturer-specific, which means that the various diagnostics and therapeutics differ significantly in terms of the allergen spectrum as well as the respective allergen concentration. It is, some extracts just lack certain allergens. In order to compensate the naturally occurring variations and to ensure consistent quality in diagnostic and therapeutic preparations, it is important and also required by authorities to standardize the extracts obtained from the raw material. If the therapeutic product contains intermediate and minor allergens, the proportion of major allergens would be too low and the therapy will not be effective, right? All allergen carriers, and thus also the raw material for allergen extracts, always contain all allergens of the respective class, but due to nature in varying concentrations. To ensure the consistent batch-to-batch quality of the therapeutics and thus patient safety, standardization is carried out during production. This standardization procedure, as required by the authorities, is administered along with the others to two target values: the total allergen activity and the quantity of major allergens. Meeting these quality target parameters ensures that allergens, which are relevant for most allergy sufferers, are always contained in the extract. In contrast to the natural allergen exposure, much higher allergen doses are required during allergen immunotherapy to achieve the targeted tolerance induction. The standardization ensures that the allergens relevant to most allergy sufferers are high-dosed, regardless of how many other allergens are present in the extract. What is the benefit of the allergen spectrum from a clinical point of view, and why is there no clinical study on it? As you have already seen in our videos on the allergen spectrum, the IgE sensitization profiles of patients are very heterogeneous due to molecular spreading. Manufacturers have attempted to take this into account by developing recombinant allergen preparations, so-called synthetic vaccines, to follow a targeted patient-tailored approach. However, due to the strict requirements of the European Medicines Agency, the EMA, it is unlikely that products for individualized allergen immunotherapy from recombinant allergens will be available in the upcoming years. This is because each individual allergen and each patient's specific therapeutic would have to be approved separately by the EMA. Clinical studies with each individual allergen are not feasible due to the high variability within the potential study population. E.g., no study participants with a comparable sensitization profile would be found with reasonable effort and in sufficient quantity. Even if the correlation between individual allergens and patient symptoms has not yet been conclusively proven, there are numerous indications from years of research on molecular spreading and recombinant immunotherapy that suggest treatment failure due to a lack of allergens. Likewise, there are study results showing that sensitization to minor allergens may also be associated with clinical symptoms. Therefore, you should pay attention to the composition of your day-to-day diagnostic and therapeutic preparations. Using an allergen immunotherapy product that contains all allergens from the respective allergen carriers, the likelihood of efficacy increases even in patients with rare individual sensitization profiles, and the probability of treatment failure decreases accordingly. If my patient is sensitized only to major allergens, but the intermediate and minor allergens are also included in the used allergen immunotherapy, am I sensitizing him with the treatment? Studies on this are rare, and thus evidence is scarce. Theoretically, it is possible that all available IET products could trigger this process, but there is little evidence of new sensitizations during allergen immunotherapy. However, even natural exposure to the anti-allergen spectrum also causes sensitization, but the contribution to the development of symptoms and progression of allergy is still not clearly understood. For an effective treatment, the patient's existing sensitizations are decisive. If these are not treated due to the absence of an allergen, the lack of success for the patient and thus the lower benefit is more serious than the low risk of a new sensitization.